Next faculty speaker is Paula Cannon, who's going to tell us about gene therapies using modified B cells.
Okay, so the good news is there's only 2 speakers left, and the one after me, Dr. Cohn, is excellent. So you just got to get through mine, okay? Right. All right, so I'm going to be talking about B cells, and we haven't heard anything about them today, so that's good. All right, it's a CIRM meeting, so I do have a stem cell on this slide. You can see here the classic slide showing the hematopoietic stem cells that give rise to all the cells of the blood and the immune system. And the reason I've put that up there is, and actually this follows on quite nicely from Dr. Shah's talk, is because increasingly we are thinking about the potential of engineering different immune cells. So here I've highlighted some of the cells that are currently being engineered. And of course, you've all heard of CAR T cells, and now you've all heard of CAR macrophages, but also dendritic cells and NK cells are also candidates for a gene-modified cell therapy. But if you look at this chart, you'll see there's kind of like a gap over on the right, and that's where the B cells are. And so this kind of, caught my interest a few years ago, and I was thinking, well, why can't we, if we can engineer T cells to make CAR T cells, why can't we do a similar trick with B cells? And the answer was, because it's really difficult, but I didn't know that then. Okay, right. So another reason why you might think about engineering B cells is because, of course, B cells make antibodies. And antibodies are the most important biological drug we currently have. Now, monoclonal antibodies are used to treat a whole bunch of different diseases. In fact, anybody who watches American television between 5 p.m. and 7 p.m. at night can see advertised a whole bunch of drugs that are based on recombinant monoclonal antibodies, right? And some of these are eye-poppingly expensive. Some of the monoclonals we use for cancer therapies, for example, can actually be $100,000 a year. So it's kind of quite an industry. So because of the value of antibodies and because of the expense and, frankly, the inconvenience of having to get multiple infusions for some of the antibodies, gene therapists are interested in the idea of making in vivo factories in the body that could be making the antibodies. And so, for example, vectors such as AAV vectors or lipid nanoparticles are being engineered to carry a payload that expresses the antibody. And typically, these are injected into muscle cell, a cell that doesn't normally make an antibody. So we thought, well, gosh, why don't we actually think about using B cells to make antibodies, because that's their day job, and any kind of complications in terms of post-translational modification is not going to be a problem if you make your antibody in a B cell. So I have a slide here that I think summarizes the other really important advantage of thinking about a B cell, which is that an engineered B cell, if we get it right, has the potential for long-term and boostable expression of our antibodies. So what I'm showing you here is we're going to do really basic immunology, okay? Here's a naive B cell, and it's expressing its antibody on the cell surface in what's called a B cell receptor, or BCR. And if a matched antigen comes along and binds to that B cell receptor, that then triggers the B cell to undergo expansion and differentiation. And you end up with 2 new types of cells. So at the top there, you have your typical antibody-secreting cell, the plasma cell, and they are now, instead of expressing the antibody on the cell surface, they now make a form of the antibody that has lost its membrane anchor and is being secreted as the antibody. And plasma cells are incredible in vivo factories, and they can live for many years, if not decades. That's why for most vaccines, once you get a vaccine once, you don't need to have a booster. In addition, B cells also make memory B cells down at the bottom, and they're poised to re-expand and recreate new plasma cells if they re-meet an antigen. And again, that's the reason why your COVID booster shots are going to hopefully work to give you more antibodies. Okay, so our goal was, can we use CRISPR gene editing to reprogram B cells to make a custom antibody? And we figured that to do that, we would need to use CRISPR editing at the actual native immunoglobulin locus in order to retain these properties of B cells that we wanted to retain, and specifically the response to the antigen and the switch from the B cell receptor to the secreted antibody form. And again, this was it seemed like a really obvious idea to me when we started working on this, because I had no idea what the immunoglobulin locus looked like. I think I must have skipped those classes as an undergraduate. But I can summarize it for you by saying the immunoglobulin locus is a gene editing nightmare. Huge locus, heavy chain, 2 light chains, multiple regions of sort of homology, and every unique B cell has a unique sequence, so really perfect for not using CRISPR gene editing. Okay, so let's stay on the basics, and I'm going to walk you through what an antibody is for those of you who also missed that class. So a conventional antibody comprises 2 heavy chains and 2 light chains that sort of pull together. And again, if you're remembering these things, the tips of these heavy and light chains, this is where the variable domains of an antibody lives, the VH and the VL domains. And the combination of these 2 together gives you the specific structure that allows that antibody to bind to its specific antigen. So if you think about reprogramming this, you would have to reprogram the heavy chain and then reprogram the light chain, which is in a completely different place. And again, there's 2 alternate light chains, so you'd have to actually look at reprogramming potentially 3 genes, so very challenging. So instead, our approach was to copy this trick that camels and llamas do. So camels and llamas, in addition to making conventional antibodies, they also make a type of antibody called a heavy chain antibody. And you'll see that I use this abbreviation HCAB, so forgive me if I say that. And so the heavy chain antibodies comprise only a heavy chain, and they are missing this domain here in the normal H chain called CH1, and CH1 is what recruits and binds to the light chain. So by skipping the CH1 domain, HCABs have no need of an L chain partner. And then this also means that because they're heavy chain only, their variable domain, the sort of the head of the antibody, is only made from a single domain which derives from the heavy chain. It's called a VHH domain. So that makes it simpler to think about substituting this region to create an antibody that would have a custom antigen recognition domain. Okay. So the next 2 slides are the 2 most complicated ones I have. So if you need to check your email, you can do it now. We've also published it. It just came out last month in Nature Biomedical Engineering if you want the details. But this is showing a schematic of the heavy chain locus, IGH. And here is where you get the variable head from. And this then splices to any one of a number of alternate constant domains that give you the different flavors of antibodies. So I've highlighted, for example, IgG1. And so what we did was we decided that we were going to use CRISPR gene editing to insert inside this constant region of IgG1. And we were going to do our insertion downstream of the CH1 domain. If you remember, that's the part of the H chain that binds to the L chain. So we're going to insert a B-cell specific promoter, the custom head we want on our antibody, and by putting it downstream of CH1, the end result is that we should produce HCABs. And because we've retained the linkage to this alternatively spliced membrane domain, then depending on the differentiation state of the B cell, we should be able to make both the membrane-anchored form of the HCAB as well as the secreted form. So that was the theory. So in order to do this, first of all, we had to decide where should we start? What should our first custom HCAB be? So my lab works on HIV, and you probably all know that we don't yet have a vaccine for HIV, and we can't cure HIV. And part of the reason for that is we don't make good antibodies against HIV. So we do actually make antibodies against HIV. If you're infected with HIV, you make antibodies, but the problem is they're not neutralizing antibodies, and the virus is constantly mutating, so it can keep one step ahead of those antibodies. However, there are some rare so-called broadly neutralizing antibodies that have been identified that resist escape. and they can suppress HIV, and they're currently used as monoclonal antibodies in the clinic, but they're only rarely made after HIV infection, and they're not at all induced by vaccines. So we decided to take one of these so-called broadly neutralizing antibodies, and the one we selected is called J3. So J3 is actually a llama HCAb antibody in its own right, and its VHH domain, you'll see over here, binds very nicely to the HIV receptor, binds very nicely to the HIV envelope protein, GP120, and it kind of neutralizes the virus in that way. So here's the experiment we did. We took human B cells, we edited them to insert this J3 head into our immunoglobulin locus. For those of you who like the details, we use CRISPR-Cas9, and we also use AAV vectors to deliver the so-called homology donor to bring in the antibody cassette. And then when we do that, we can take our human B cells and we can look to see whether or not we've created J3 BCR on the cell surface, and that's shown here. Yes, we have. And then if we take those B cells and we differentiate them in vitro towards plasma cells, the antibody secreting cells, we can also see that we get secretion of the antibody into the supernatant, and that supernatant can neutralize HIV. Okay, so that's a cute trick, but do our engineered B cells do the thing we really want them to do, which is, do they respond to their specific antigen? Do they respond to immunization? So this is where things get a little bit difficult, because it's quite hard to study human B cell function in an animal model. We could work with mice, but if we work with humanized mice, they don't fully support human B cell function. So this was problematic for us. And then a couple of years ago, a nice model system was developed called the tonsil organoid. This came from Mark Davis's group. And what tonsil organoid models are is they're like a little human immune system in a dish. You take tonsil tissue, you pull out the B cells, we can engineer the B cells, and we mix them back in with the rest of the immune cells that are present in that tonsil, and then we can expose them to our antigen. In this case, it's GP120. And what we see when we do that is that we see this nice increase in the number of GP120 specific B cells, but only in the case of the GP120 vaccine, and only when we've actually added in our engineered B cells. So this is how we test whether or not our B cells are responding to their antigen. Okay, well, that's great. We've done it in a dish. Do our HCAb-edited B cells work in vivo? And again, as I've already explained to you, the problem is we can't do this in a humanized mouse. So we had to go into non-human primates, rhesus macaques. And so that kind of put us back for a couple of years, because we'd done everything in human B cells up to this point, and then we had to kind of start from step one again and figure out how to do everything in rhesus macaque B cells instead. But we were able to do that. We've done one pilot study. I'm going to show you the data on one slide next. And we're currently in the middle of our second study, but I don't yet have data on that. So let's take a look at what the approach we took with these macaques. So first of all, I just want to point out something that's actually quite nice about HCAbs compared to conventional antibodies. So again, remember, we're putting in this little custom head. This is the orange box. And so far, I've shown you we can do this with a VHH domain. This would be J3. But we can do it with other things. So for example, we can do it with a single chain antibody. We can put in a domain of a protein. It doesn't even have to be an antibody. And we can multiplex and make all sorts of very non-natural molecules using our imagination. So for our rhesus macaque studies, we decided to choose two separate HCAb designs, and these were made with a collaborator, Wei Li, at the University of Pittsburgh. So first of all, we took J3, but he humanized it for us, so we thought that might reduce any antigenicity when we put it into the macaques. And we also worked with a single-chain antibody derived from a very famous BNAb, famous if you know about BNAbs, called PGT121. Okay, so let's take a look at what happened when we put these into macaques. And the schematic is basically we take blood from the macaques. We only do a small bleed. We take about 50 to 70 mLs from the macaques. We purify out the B cells and we engineer them. And then we return them to the macaques at the same time as we vaccinate them with the GP120 protein. And then we follow up at 4 and 8 weeks with additional boosts. So here's the data slide for this first experiment. So first of all, on the left, whoops, oh no. Oh, figured it out. Okay. On the left, these are 2 animals that got the humanized J3 HCAB. And you'll see that this is the limit of detection, that we did actually see HCABs in the serum that lasted out to about 10 to 12 weeks. And then over on the right, you see the same experiment. This time, there were 4 animals that got the PGT121 SCFV derivative. And of those 4 animals, only 2 of them we could detect the HCABs in the serum, and with one of them, this one in red, it did actually look like the animal responded to vaccination. And as I said, we have a second experiment now ongoing with macaques where we've massively increased the number of B cells we've put into them to see if we can increase that signal. Okay, so to summarize, hopefully I've convinced you that we can use CRISPR gene editing to reprogram B cells for the production of custom antibodies that respond to their antigen. And our pilot NHP studies have detected these HCABs in 4 of the 6 animals receiving engineered B cells. And then just thinking in terms of what could we use this for, what could be the applications? Well, I think, as I've said to you at the beginning, this has the potential to provide a long-lasting source of antibody, or actually non-antibodies. We've done this with non-antibody proteins that can be secreted in the body and have the potential to be boosted if the levels start to decline over time. And HCAB editing flexibility, remember, we can swap whatever head we want to put on. This supports us using completely non-natural, non-antibodies and anti-self antibodies, such as those that could have applications against viruses, against cancer, and against autoimmune disease. And with that, I will just finish and acknowledge the current and previous members of my lab who are involved with this and our great collaborators and sources of funding, which don't include CIRM. Just thought I'd mention that. Okay, thank you, everybody.
Incredibly interesting talk. Did you measure or compare relative expression of wild-type antibodies versus your engineered wild antibodies in vitro or in vivo?
Yeah, that's a great question. Yeah, we did. So when we engineer our B cells, one of the things that's kind of interesting that I didn't mention is we're not actually preventing expression of the endogenous antibody. So we often get 2 antibodies expressed, and we can detect our antibodies versus the endogenous antibody, because the endogenous antibody has a light chain. So when we measure the 2 together, we see very comparable levels. Yeah.
Thank you.
Thank you. Okay.
Hello.
Hello.
Beautiful talk. Thank you. I had a question about the CRISPR-Cas9. I know that you mentioned that it's easier to do that with just the heavy chain, but now you run the risk of losing specificity without the light chain. Did you observe any differences in vitro or in vivo because of that?
Okay, so we don't lose specificity because if we were putting in normal antibodies, we would need heavy chain and light chain components. But when we put in, if you remember the molecule I call PGT121, that's actually a single SCFV. It's a single chain antibody. So it brings in both the light chain and the heavy chain variable regions, but we stick them both on the sort of the top of the H chain. So we retain it in that way. And then, of course, we can put in other types of what we call, the head. We're not limited to the natural heavy plus light chain combination. And the J3 VHH domain that we use has exquisite breadth and potency against HIV far better than any conventional antibody I've seen. So I think there's a lot of potential to make different types of antibodies, and so far we haven't seen lack of specificity as a problem.
Thank you.
Thanks. Okay. All right. Thanks. Thank you, everyone.